students. Today we will discuss about a very unique and the most important part of molecular basis, that is gene. What is gene? Yes. Now it is a very difficult to say what is gene in one word, clear, or in one sentence. And we know that concept of gene is never a static one. It is gradually, regularly, it is getting refined. So let us discuss how the gradual change in the concept of gene or gradual evolution in the concept of gene occurred, and now how we can define gene. So students, normally we define gene in various ways, clear. Now sometimes we call it some sequence of nucleotides that which is DNA. Sometimes in viruses it may be RNA, which encodes for a synthesis of a protein, or it may encode for synthesis of an RNA, clear. And that is a segment of chromosome which is heritable. And so this is the in gist concept of gene. So what is the concept of gene? It is the sequence of nucleotides. That means whether it is a deoxyribonucleotide, or in certain viruses it is a sequence of ribonucleotides only, clear. And which this sequence, what is meant for, what for it is meant? It is meant for synthesis of some proteins, or sometimes synthesis of simply RNA without proteins, clear. So synthesis of RNA or synthesis of protein. And another unique feature is that it is heritable, so it can be transmitted from one generation to next, and overall it is a segment of chromosome, clear. So this is what somehow we can summarize the gene. Now we will see that how this concept of gene is changing with time. First of all, Johansen was the first to coin the term gene, clear. And after that, Biddle and Tatum, they proposed that gene is a segment of chromosome that is responsible for formation of one enzyme. It contains a sequence coding for an enzyme, clear. So one enzyme, one gene, one enzyme theory by Biddle and Tatum. Later on came Fusky. He told that it is not that one gene, one enzyme, because many a times we see that a single polypeptide is a constituent of various enzymes. So it is basically one gene, one polypeptide, clear. So Fusky told it is one gene, one polypeptide. After that, Benzer proposed that a particular segment of DNA or particular segment of chromosome that is responsible for the formation or sequence which contains the sequence of a particular polypeptide can be coined as cistron, clear. So he proposed the term cistron, which is a segment or sequence of deoxyribonucleotide which is responsible for coding for a particular polypeptide. Now next, what is seen? Next it is seen that one gene, one polypeptide, or finally the term cistron came for one polypeptide. Now the question is how it is controlled. Now when this polypeptide needs to be synthesized, how this control mechanism is done. Now to find it out, it was seen that this is the part which is coding for a polypeptide, but there are other parts also in the DNA which are regulating when this segment, that particular segment, is to be coded, clear. So that part, that is also a part of that chromosomal DNA, clear, which is regulating this particular segment when it is to be coded. So those are the regulatory part. So now the concept of gene is extended to not only to for the formation of polypeptide but also to regulation of the formation of polypeptide. Clear? So now the concept of gene is extended from not only the to formation of a polypeptide but also the certain segment is responsible for regulation of this formation of polypeptide, when is to be formed and when is not formed. Clear? So that portion is included. Another thing is also seen that sometimes in many cases, like suppose tRNA, the RNA, this is coded from the DNA only, but this tRNA is not translated to certain proteins. So there are certain segments of DNA which only codes for a RNA, wherefrom only RNAs are formed, but they are not translated to polypeptides or proteins. So in a broader sense, gene includes this regulatory part also. Clear? So the concept of gene is extended to a regulatory part also. Then the part which is coding for the polypeptide, that is particularly called structural gene, and another part which doesn't code for a polypeptide but actually is transcribed to RNA. Clear? So all these are actually in the broader extended concept of gene. So now we can call gene as one gene, one function. Not one gene, one polypeptide, not one gene, one enzyme, but basically the structural, the part of the gene which is coding for a protein, that is polypeptide, which may form enzyme or other protein bodies. There are other part of the gene which is regulating when this particular protein is to be formed. Clear? Another part of genes which is not forming protein, they are forming only RNA. So that the extended concept of gene is one gene, one function. So suppose this is the double-stranded DNA. Clear? So suppose this part is the part which is actually coding for a protein. Clear? Or a polypeptide, basically a polypeptide. So this will be first transcribed to RNA and then it will be transcribed to a particular protein, translated to a particular protein. And there is associated with it, there is another part that is also a sequence of DNA, clear, deoxyribonucleic acid, which is responsible for regulating this part. Clear? So this particular part which is responsible for coding for a particular polypeptide is called structural gene. According to Benzer, it is the cistron. Clear? And that particular part which is not coding for a particular protein but it's controlling the structural gene or the cistron. Clear? This is the regulatory gene. So in a broader concept of gene, we consider all the part. Clear? Not only the part which is coding for a protein but also the regulatory part. So we are including all this part as a gene. So, and sometimes this structural part is not coding for a particular polypeptide. It is coding for a particular RNA which will never be translated to a polypeptide. So that is also within the concept of modern gene. So it is a broader sense we can say that one gene is responsible for one function. Again, another thing is that suppose this is the regulatory part. Clear? So suppose still this is coding for a particular protein. Beyond that, it is coding for another polypeptide. So this is the regulatory gene. So till this is coding for a particular polypeptide. Clear? Next, beyond it, it is coding for another polypeptide. So it may happen that under one regulatory gene, there may be numerous cistrons. So suppose this is the part which is coding for one polypeptide. There may be another part which is coding for the next polypeptide, but the regulatory gene is the same. So that is called polycistronic gene. clear, which is common, very common in prokaryotes. But in eukaryotes, generally, one regulatory gene actually regulates for a particular cistron. So most of these eukaryotic genes are monocistronic. That means one regulatory part or regulatory gene regulates a particular cistron coding for a polypeptide, not multiple cistron. But in prokaryote, we've seen that one regulatory mechanism clear is controlling multiple cistrons, numerous segments, which is responsible for coding for a numerous polypeptide. So that is called polycistronic gene. So we have learned what is polycistronic gene, clear, present in prokaryote, one regulatory mechanism controlling numerous cistron, and what is monocistronic gene, which is present in eukaryote, one regulatory mechanism controlling only one cistron only. Now, in eukaryote, we find another thing also, that suppose this is a cistron. Clear? This is coding for a particular polypeptide. Now, it is seen that sometimes all these are not coding for a polypeptide. Sometimes there are numerous nucleotides which are not coding for the polypeptide, but they are present within the gene, within the structural gene or within the cistron, but they are not coding for a polypeptide. Clear? When this polypeptide will be coded, will be formed, then this particular new amino acids or this particular nucleotides will not be taken granted. Clear? So that will be spliced off. So this type of gene, when there are certain sequences of DNA which are not which is not the part of the polypeptide, but they are in the cistron, this type of so they are intervening the normal functional part. So these are called introns, and this type of gene which contains introns are called split genes. So what are split genes? Split genes are those genes which contain some non-functional nucleotides in between the functional nucleotides of a particular cistron. Clear? So split genes are those genes which contains certain non-functional nucleotides within the functional nucleotides of a cistron. So this type of non-functional nucleotides are called introns, and this type of genes are called split genes, and the functional nucleotides are called exons, and these are seen generally in eukaryotes. So another thing is that sometimes we see that certain genes are always functional in a cell. Certain set of genes are there which are always functional in all cells, like the genes which are responsible for our respiration, formation of the enzymes responsible for respiration. Clear? These are functional in all cells and always they are functional. So this type of genes are called constitutive genes or housekeeping genes. Clear? Which the set of genes which are functional in all cells. Clear? Always functional in all cells. So they are called housekeeping or constitutive genes. Now certain set of genes are there which are functional at certain times in certain cells. So these genes are called luxury genes or non-constitutive genes. So constitutive genes are those set of genes which are present in all cells, which are not present. Obviously all genes are present in all cells of a particular organism. Clear? But which are functional in all cells always. So those are called constitutional genes, clear, constitutive genes or housekeeping genes. And certain genes are there which are not functional always, but they are functional when they are needed. So this type of genes are called non-constitutive genes or luxury genes. Now what is genotype? Genotype, we know it is the genetic makeup of an organism. Clear? Of the cell, of a particular cell. Genetic makeup of a particular cell is called genotype. Now these genotype is actually responsible for the transmission of the characters into the offspring, and finally the formation of the particular phenotype. Clear? So this particular genotype, that is particular genetic makeup, is responsible for formation of a particular phenotype along with the environmental and other factors. Clear? Now another term is always often heard in this concept is allele. Now what is allele? Now certain genes are produced. How these genes are produced? Certain genes are produced. Generally genes are by any mutation or anything it happens. There is a change in the DNA sequence or change that may form a particular character, and this particular character, by if it is selected by nature, so by natural selection, finally it becomes a particular gene of an organism. Clear? Now sometimes it happens, suppose this is the gene. Clear? This is the particular sequence which is responsible for forming a particular polypeptide, and that is naturally selected, and now it is the gene in that particular organism. Now if by certain mutation or by any other factors, suppose these nitrogenous bases, clear, certain nitrogenous bases alters, then what will happen? If there is any change in the nitrogen base, then there will be change in the coding sequence, and as a result there will be change in the protein amino acid, and finally there will be change in the protein. So there will be first, if there is any mutation, there will be change in the coding sequence, and this will change the RNA, this will change the amino acid, and finally the protein, and so it may alter the phenotype. Clear? So these are called alleles. So these are called the alleles, that is alternative form of a gene. Clear? That's a called allele. And we have already learned that it is not that always DNA forms the gene, because certain viruses, RNA is the part which is forming the gene of that particular virus. So we can see that concept of gene is never static. Clear? By the time it is going on regularly, it is going on refined, and so now we can define gene as a discrete locus. That means a particular locus, a particular point in the heritable locus of heritable genomic sequence. Clear? Is a particular point which contains a sequence which is the DNA sequence, or in viruses it may be RNA sequence, and which is heritable. That means it can be passed to the offsprings, and which an organism, clear, trait by being expressed, which is expressed directly as a functional product, or it may be a part which is regulating that particular formation of that particular product. So how will you define gene? Gene is a discrete locus of heritable genomic sequence. So it is a particular point on the genomic sequence, clear, which is heritable, which an organism trait, it is reflected by a particular trait of that organism by coding for a protein or by regulating that formation of that protein. Clear? So thank you, students.